The overall goal of this confocal live imaging method is to directly observe the cellular processes that drive secondary palate fusion during craniofacial development.
This method can help answer key questions in craniofacial biology field, such as what are the cellular mechanisms that regulate lip and palate fusion. The main advantage of this technique is that we can directly observe cellular process of tissue fusion at the cellular level in real time.
Begin by separating the head from the body of the embryo under a dissecting microscope in a petri dish containing fresh, room temperature PBS. Grasp the head with one pair of fine number 5 forceps, and use a second pair of forceps to remove the upper brain, cutting away any extra tissue outside of the palatal shelves with the second pair of forceps. Remove the mandible carefully without damaging the palatal shelves. When the mandible has been detached, carefully remove the tongue and confirm the presence of a robust reporter signal in the midline epithelial seam.
It's important not to damage palate explant during dissection to keep the midline epithelial seam intact.
Next, remove the hindbrain, brainstem, and both maxillae with the adhered palatal shelves. Cut the anterior upper jaw tissue, keeping both the primary and secondary palates intact.